Hello.
Hi,
How are you?
good, good.
Great. Hello, Jesse. All right, let's get started. That's got a little sparser group here today, if that's okay. This was another good week. We did DevaWorm, of course. We had another DevaWorm meeting. And then we did Cognition Futures. We finally got back to Cognition Futures, where we finished up the allostasis paper that we were working on before. So I don't know if we want to give any updates now or.
Sure. I mean, a lot of good neurotech developments, which has been really great to see. And yeah, there's a for those interested, there's a new paper out on MOAB, which is Neurotechnics' mother of all BCI benchmarks. That's great to see. I think there's real opportunity there for those interested in the kind of machine learning side of capturing kind of early in this case like motor imagery or kind of clear psychophysical representations in steady state VEPs and things like that. And some good also follow-up meetings in terms of some of the work that's come out of the Pickhour Institute in Boston on 40 hertz stimulation for, I think the putative physiological mechanism here is improved lymphatic clearance.
Okay.
In terms of both reduced clearance is what's building up in dementia in terms of malformed proteins or proteins that you don't want, it's just interesting that 40 hertz stimulation might have this kind of non-neuronal action. It's not neuromodulation that you're doing this for, or in this case it's actually just stimulation, visual stimulation, but that it would have this this physiological
effect
response. Effect is pretty interesting. And next week is the BCI Spring School. I forget now what they call it, but GTech runs this. It's a company based in Austria. And it's definitely supported and marketed by IEEE Brain. But it really is an incredible free course. So many speakers. I think it's got 40 plus speakers. So it's running from something like April 22nd to beginning of May. And the worst spot to be is in the West Coast.
Why is that?
Because of the time zones.
Oh.
like we kind of have the worst of the time. I mean, I haven't worked it out for something like New Zealand or something.
Right.
The presentations be a lot of really interesting applications and people applying this, and then they're doing a hackathon too. It's great. And I think that's about it. Or, check the Slack. There's definitely some interesting things that are going on in the world too, or, interesting papers. And I'll have a look through.
Okay. Well, that's great, Morgan. Thank you. I saw reference to that 40 hertz paper you're talking about, and I don't know if you remember the binding literature, but that was where they hypothesized that 40 hertz was doing everything in the brain.
Right, because of gamma. Yeah. Right.
So that's interesting. Yeah.
Yeah. It is. I mean, I think that was an early idea, was that somehow you were stimulating gamma, but I mean, certainly, I think the first review paper on glymphatic system was like just a couple of years ago. Anyway, it's interesting because the guys in Norway at Simula that developed Phoenix, the finite element package, when I was put in touch with them to talk about what we were doing with electrical impedance tomography, I said, so what are you using it for? Because somebody had told me he's using it for brain stuff, and he was like modeling the lymphatic system. Oh. And I was kind of like, well, what's that? So, but he was the one who told me, you can get dementia with just a case of CSF unable to clear, and your head fills up with fluid to the point where it's basically there's so much pressure on the brain. But now I wonder, is it pressure, or again, was it actually a case of clearance?
Right.
Anyway, super interesting. Check the review papers on glymphatic system, because it's definitely involved in the immune system and its complexities too.
Oh yeah.
Which again has this very kind of narrow network of capillaries, or I don't know what you call the ducts.
Yeah. I'm not familiar with the glymphatic system.
Yeah.
I saw the intro to the paper, and they said it's a relatively new system. So I don't know when it was kind of.
Yeah. And with the kind of deep tech VC company here in San Francisco, have been kind of following interesting companies have been focused on that, where either they're doing surgery to kind of open more holes, crudely put it. I'm sure the surgeon, I hope the surgeon says something more technical. We're going to perforate your glymphatic system.
Yeah.
But it's sort of a surgical option, and I've seen some MR looking at it, trying to gauge it non-invasively. So I think it's really interesting, because there's been a lot of renewed research in other Alzheimer's treatments now that the stranglehold of the paper got us started on the particular protein seems to have had some falsified data.
Yeah. About the plaques and things like that.
Yeah.
Yeah, that was like the dogma of that area.
Right. And I remember researchers a couple of years ago had a big open letter, saying like, this has had a stranglehold on research, and there's a lot more going on, and And I wonder, some interesting use of organoids recently, looking at COVID's immunological effects, that I thought was an interesting application. And I wonder how much again, like the the kind of, the renewed research in COVID and long COVID is changing the medical scene about the complexity, especially around the brain, in terms of the viral action.
Yeah. Hey, Paula.
Hey, Paula.
Yeah, that's maybe we'll go over the brain organoid stuff today. The channel. Well, we haven't done that in a while.
Pasca's got a new paper. Pasca Lab in Stanford's got a new paper. It looks super interesting. I mean, I just saw it last night, so I haven't read it.
Yeah.
And this week has been their the brain organogenesis workshop, which is Stanford's. It's the course where I want to be able to see their course material.
Yeah.
I want to know what equipment they use and you have I have reservations. Oh, you have reservations about that work. Okay, let's dive in about. All right, let's go.
I don't have space because it's late night here, and I turn into a monster at night. So, can you hear me?
Yes.
One of these days, I will make myself presentable for the meeting. But I just remember that this is about almost 11 p.m. and I was already passed out. And then I said, maybe this is fine. So thank you for reminding me. But I have reservations because of the eyes. Only the brain organoid, the brain organoid in particular that develop the eyes. You know that, right?
Yeah, absolutely. Yeah.
So, and that says something, doesn't it? And so I just thought, I made a joke when I heard about the eyes, and I thought next they're going to develop a mouth, and they're going to make inquiries as to who's given the ethical review for this year. And my reservation is we don't know. We don't know. I didn't want to say much. I didn't say take away the enthusiasm, but we don't know enough, I don't think, to start developing brain cells and assuming that they're we don't know what kind of consciousness. But it's not just me. I presume there's a lot of people who have reservations. Is that right?
Well, if you're, you might be referring to the early work by Bradley Voituk and Alison Moultrie, where they were showing oscillations in the organoid after a certain amount of time.
No, then the organoid, the cell develop eyes.
Oh, yes. I mean, you can grow. I mean, certainly there are retinal organoids, and some organoids where people are like trying to stimulate while the organoid's growing.
So I don't know enough. But when I heard that, let me put it up. I don't know what kind of organoids they were growing. I think they will. I remember that being brain organoids.
I seem to remember that too. I remember that too. And when Alison Moultrie came to speak at the Exploratorium here in San Francisco, which is a kind of famous science museum, he talked about the internal debate that they had when they started seeing, in the electrophysiology, they started to see, kind of neonatal-like power spectrum in the electrophysiology, and there's certainly questions of, it's such early days. Do we know what we're growing?
The article that I think was by W. Liu, which says we report the self-formation of human cephalon eye organoids comprising concentric zones, and it was like a spontaneous thing. They didn't prompt.
Yeah. I know the paper you're talking about. you know, and yeah, that was.
And so
like
that kind of, I mean, that kind of was important observation. I mean, an important thing, because it showed that we don't just develop things as we plan to develop them; they develop by themselves the way they.
Yes, I mean it's a perfect. I mean, I think of it as such a clear demonstration of the kinds of things that Michael Levin's talking about in terms of that. You know, DNA isn't this. You know, it isn't a set of architectural plans. It's the coding for a number of proteins and kind of different inhibitions and you know promoters of said proteins and
So people are continuing to develop brain organoids, and you know, I think maybe there should be some ethical issues addressed. How I don't know much else really. So is it really useful to do to develop the organoid?
well, so you know, for specific questions, yes, right. But I mean, but very specific questions, and with an acknowledgement that this is still very much in vitro experimentation as opposed to, you know, live animal, you know. But I think it's you know, one, there are groups of people who are looking at organoids as basically a non-animal testing platform, especially for toxicology, right? So you know, and if you know, if you know cancer research, like the number of animals that are used in cancer research is you know, tremendously huge, and so again, like I absolutely understand the ethical concerns that there is this thought though that this is still better than working with animals, as well as, you know, in one respect limited, because again, the gene expression and the tissue organization and development isn't natural. But at the same time, you know, I've seen some really interesting talks and papers of people saying, you know, that some of these even kind of basic assembloids and spheroids can be, you know, used better and at larger scale than animal testing for pharmaceuticals, for instance, you know.
Let me just say one thing as a broad point. I have reservations about how medicine goes in general about reviewing diseases and addressing diseases. I have reservation about a bunch of bioengineering approaches to. Now, the last thing I read, which was kind of for me was a bit shocking, was they were merging the DNA of the tardigrade with, was it with the human? I don't know, something like, for me it was like, oh my god. I can understand, right, from a scientific viewpoint, biotechnology and bioengineering have provided a very fun playground and very interesting scientifically, because there is virtually no limit to what scientists can do in a lab with quite limited equipment these days. But what I lament is that there is limited awareness of a lot of scientists as to whether, you know, we could be using our resources better, and whether if we understood the nature of diseases. So we can spend a lot of money and time trying to study cancer at genetic level, but maybe the causes of cancer are to be found elsewhere. like lifestyle, but also there are a number of hypotheses that emotional trauma can cause genetic mutation, that kind of thing, yeah. And so that there are a number of things that make people sick. Nutrition and other chemicals in the environment and radiation and a number of other things. So what I'm saying is that there is a lot of emphasis and a lot of resources put into
absolutely.
research at molecular genetics
Help,
which is fine. I can understand why, but I'm not sure it's the wisest. I mean, I don't agree with that line, and of course, there's nothing I can do about it. I can only go about it a different way myself, which is what I do.
Sure. One of the papers that I put in computational psychiatry this week is focused on kind of the physiology of trauma.
You want to share do you have a preprint or something?
So in the Slack, I'm going to sneeze again. Okay. In the Slack, if you check computational psychiatry, it's the 1 second. Towards understanding and halting legacies of trauma.
Okay.
Yeah.
Thank you.
Yeah. And as I've talked about before, one of the clearest findings in psychiatry, when you set aside the question of fuzzy labels or even labels that don't even potentially make sense, is the actuarial data in terms of patients who get a diagnosis of whatever sort of major mental illness are going to die younger, and by 7 to 10 years, and understanding the physiology of that, and perhaps targeting that physiology would make more sense as kind of a healing than say the kind of symptom management that we focus on. Just as an example, but just wanted to say one other thing about organoids that I think it's really interesting to look at what Michael Levin's saying about the role of bioelectricity in spatial morphogenesis, and having a way to look at that in mammals is one of his new directions as his morphoceuticals company, and that really does the goal of that would be to bring regeneration to mammals, or say limb regeneration, right, things like that. And potentially even organ regeneration, right, would be also a kind of long-term goal of that, right? So heart disease is still I think heart disease is the #1 killer. It's definitely diseases of the heart rank up there with cancer in terms of I think numbers. And
cardiovascular.
But heart disease has been targeted because, or I should say, heart organoids, cardiac, or would you call them cardiac organoids?
Right, cardiac organoids.
Is people are talking about growing organs for people. Again, certainly is a really I'm glad I live in a time where we can start to tackle the biotechnology around that. But with an understanding of all the caveats that you've brought up in terms of ethical considerations.
Of course, I understand that from a scientific viewpoint, it's very interesting, and having the instruments and having it, and so one has to do it. But at the same time, I take maybe because I'm not working in that field at that level. Sometimes I wonder, whether prevention or other things. I think there is too much emphasis on the speculative or theoretical research, which is interesting, very interesting, because we learn about the world, about the universe. I'm not saying it's not good, but we don't have. There is not enough information about how to stay healthy out there. For example, the medical care doesn't teach you how to solve problems before they arise and all of that. That's the space where I am. Where I'm at. Anyway, I've got your paper. Thank you, Jess. I'm going to download it and read it.
Yeah, good.
Yeah, so that's great. Thanks for that conversation. And just to throw kind of a bomb into this, maybe, or now in DevoWorm, we've talked about organoids, and we've come across this literature on human embryoids. So people are actually trying to build
bomb.
that's a huge, if you're interested in the ethics of it, there are people who have developed small, kind of very early human embryoids, and there's been a lot of people asking ethical questions about that, saying, what is this? Is this like human, like a very tiny human? Yeah, there we go. Or is it, what does it resemble? And it's the question's still out on that. You get to a point where you can do things, and you have to ask some pretty serious ethical questions as to where, if we're actually creating humans in a dish. And a lot of the embryoid work, of course, or organoid work, you get sort of onto that boundary where you're pushing a boundary where you say, is this like a human system or not? And we say, well, not really, but it could exhibit some aspects of human systems. So there are, if you're interested in the ethical issues, my point is there are a lot of interesting ethical issues in some of these emerging technologies, and if we're interested, we could talk more about it. But it's something I don't think people haven't really tackled too much yet because it's such a new technology. So
yes, I'm interested, but I think I'd like to. I don't know whether it is in the scope of this group's, but the question for me is the ethics of science as a whole, because it's a bigger issue.
Yeah.
I'm just saying that there is free energy, okay, from waves, from sun, and we're still putting a lot of research and focusing. The economy is still centered around carbon or nuclear. So what I'm saying is, because the science is serving a purpose to support a certain type of, say, world order, and until that happens, even the health and medical research will follow that, trying to focus on disease rather than the prevention of the disease and all of this. So I think ethics, yes, important, but I think there is a need for a very big shift toward using scientific approach and tool towards a more positivistic, in a more positive direction. Like, let's focus on the solutions rather than the problems in that sense.
Yeah. Okay.
Until that happens, until people understand that is where we put the energy, that's where the stuff is going to grow and develop. So if we continue to put energy on understanding all the possible forms of cancers and all the possible mutations, which is interesting, and we're not going to put the collective energy of scientific establishment towards prevention and having healthy environments and reducing traumas, which there is trauma everywhere. Now I'm just starting to read this paper and talk about past events, but we have traumas in our daily lives wherever we go. And so there's, and until we understand, scientific establishment understand that, there are certain factors which are actually causing all these diseases, and maybe, so the ethics should be more like a foundational issues of where we are putting our energy and can we put our attention and resources towards to focus all these nasty things not to happen at all, if it's all possible, or to limit them or reduce them, rather than completely forgetting about what is causing them and just putting all our resources to study the beauty of the mutations and the depth of the, how far can we go into the molecules and into the matter, and because we can get lost, we get lost, but we never finish understanding the world at that level. There is too much of it, and so that's what I'm saying. The ethics, I'm trying to say that I'm interested in ethics, but I think ethics should be foundational, and it should address the laws of science as a whole, and then we have issues versus energy issues, and other issues will follow and fall into that. That's what I think, or I hope.
Yeah.
I agree, and I know we may want to talk about other things soon, but I'll very, very briefly pop in and say, yes, and a long-term, let's say, interest of mine that's more than interest is understanding where in many different fields the pressure is to kind of maintain a certain pathway versus where other pathways can go. And pretty much based off of what you said and why both the range of opportunities and the range of pressures that are sort of consolidating things outside of just obvious things like, ooh, money or funding, but also like the inertia in the different fields. And that's definitely something that I think is worth looking into. There was a paper one of my friends posted somewhere something like, this person thinks that philosophy of science should be taught to undergrads, who are undergrad STEM students. And I was like, I agree with that. And then someone else said, who had due respect, was like, well, I think that's true, but you got to be careful because you can't put too much like politics and activism and how you're teaching philosophy and science of history, because then it's just going to be a mess. And it's like that it dovetails interestingly into this comment by a, let's say, well-known technological commentator that popped up this week too, who was kind of admonishing, well, tech companies just don't talk about anything else, just do your work and do tech, do really good tech, and that'll change the world. And I mean, I agree with that to an extent, but I also feel like we're kind of in it. There's no perfectly beautifully abstracted person just doing something, even if they morally choose to not engage with stuff and want to come from a pure standpoint. Yeah, that's a whole. I don't want to open up a big old can of worms to talk about that the whole time, but I think what I constructively want to say from this is I do think that having real discussions about the many influences at play, both internally to the discipline or science or whatever, and externally. And by internally, I kind of mean paradigms and players and methodological limitations, techniques, and then also, the things that are actually going on in the world that are affecting it. Science doesn't happen in a vacuum kind of stuff too. And I think that's just not, I think there's a lot of work there to like, how do you do that in the right way? How do you do that in a sober way? How do you do that with the many, many, many pressures that are at play? And I think that's, suffice to say, I have an interest in I have some projects that are on that long term, but I mean, it's not like I don't have my fingers on the pulse of all these industries and big AI and big tech to do anything about it, but it's something worth thinking about for sure. So I'll leave it at that.
Yeah. Paula said, thank you, Jess. Yeah.
Also, hi, Adama. Excuse me.
Yeah, no, I think that's what kind of why we have these meetings, is to have good discussions. So, thank you for that discussion. And I think that we always have something to follow up on too. So, we'll probably no doubt think about it more, and we'll probably come back to the organoids channel later, and maybe we have some. I don't know if we'll answer any of those questions. I doubt it, but still, we'll we'll see. I mean, it's So Adama, hello. Yeah, so Adama wanted to make a presentation, and I promised her last week that she could do that. And so Adama is a GSOC applicant, and she's interested in the VR project, virtual reality, augmented reality, extended reality. And so, are you ready to go, Adama? I can unshare my screen, yeah?
Yeah, thank you very much.
All right.
I will share my screen.
Yeah.
Just a minute. Okay. Okay, can you see my screen?
Yes.
Yes.
Great.
You see my. You see the title?
Yes.
Yep. Copy focus and flow state.
Okay. So, welcome to my presentation. Today I will talk about my project proposition for the GSOC. I want to study the hyperfocus and flow state in a virtual reality context in case of learning. So first, I will introduce you what is flow and hyperfocus, and which are two states of high concentration level and high productivity. So hyperfocus is a state where you focus on one single task and you forget everything around you. Flow is a state where you concentrated, but you do everything effortless and naturally. You are completely involved in what you are doing, and there is like a serenity feeling. Some people describe it as the zone. So as I said, it includes productivity and creativity. And hyperfocus is often associated to neurotypical people. For the flow, it's not associated to a neurological scheme, but one thing to add is that time pass is hypnotized. So there is some internal characteristic and external characteristic. Here I will talk about the internal characteristic. For hyperfocus is, as I said before, intense concentration on task. There is like a commitment and a satisfaction while doing something. There is the loss of time notion and the loss of consciousness of environment. I describe hyperfocus as a double-edged sword because there is some more negative aspects, like difficulty to enter the state, and it is almost an obsessive state, and it can lead to mental fatigue. For flow, you don't have this unfavorable aspect. And you have the positive aspect of hyperfocus, plus you lose the loss of self-consciousness. There is a height sense of control. Everything feels effortless and enjoyable, as I said before. The experience is autotelic. It means that the experience in intrinsically rewarding, and there is a merging of action and awareness. That means that you do things without really have the conscious to doing the thing, and there is like happiness and well-being while doing a thing in a flow state. For the external aspect, hyperfocus are mainly triggered in distraction-free environment when there is like some cognitive challenges in a calm environment, and often it's a trigger when there is like a pressure, for example, the pressure of the milestone. For the flow, there is it's trigger when there is a high equilibrium between challenge and skills. The users, the participant of the person who experiences flow has a sense of control. The goal has to be clear, and there is an immediate and unambiguous feedbacks. We can observe hyperfocus in a wide range of activities, like gaming session, working on project, learning, or other thing. And we can observe flow as the same range of activities of hyperfocus, but we can also observe flow in not high cognitive challenge, like housework or, I don't know, washing dishes or other things like that. Or is it one thing interesting is that an hyperfocus state can possibly lead to a flow state. So a little focus on flow. As I said before, this is an equilibrium between skills and challenges. And when you have this equilibrium, you are in the flow channel. The first graph is a previous one, and the second one is a more recent one. I think the second one is more related to the actual research. Okay, so now let's talk about virtual reality. So virtual reality is helpful for facilitate to create a distraction-free environment, to create a higher concentration environment. It provides grounds for autotelic experience and immersion. It's easy to create a wide range of environment and tasks, and it's appropriate for distributed research since you don't have to have someone to see someone in a specific place and observe him. And some research say that their learners perform better than traditional learners. Okay, so the purpose of the study is to develop a VR learning environment to allow to assess and augment hyperfocus, augment the probability to see hyperfocus and flow state on people. So for that, I want to create a customized VR platform, create some specific task which is likely to trigger flow or hyperfocus according to the external characteristic I presented you before, and create some method to detect hyperfocus or flow. Okay, so here I describe quickly. For the task, I thought about some coding tasks for beginner and gamify task. and create a scenario, and then this task has to be in an environment that is likely to trigger a high concentration state like hyperfocus or flow. And there is some detection method that literature use, such as experience sampling methods or flow state questionnaire. And I think I thought about some other one like post-learning quizzes. Put some random signal that the user has to stop or has to click on to stop it, to know if he is to have an educator about his engagement. We can also track the movement of their headset, and we can track the time the user takes to solve an exercise. For the task, I have some inspiration. There is the MIT APP Inventor, that is like some blog that user has to put together. There is Anguera and France IOE, which are some platform to learn coding for very beginners. I used it when I was younger to learn how to code, and it's a gamified way to learn how to code, and there is like a scenario, and I think it would be interesting to implement this in their context, for the context of assess hyperfocus and flow. For the environment, I have some inspiration, especially virtual desktop, which is a platform to stream the computer screen on virtual reality view. And I think that it's interesting because we don't have to, the environment is quite simple, and I feel like that this environment is helpful to enhance concentration. For the detection methods, as I said before, there is the FSQ, so some questionnaires and a quiz. Here the Goria, invisible Goria test. It is like what I described before, the signal that I want to show in the VR context, to know if the user is focusing or not, and track the VR headset and the behavior of the user. To do this study, I will use a MetaQuest 2 because it's cheaper and it's easy to get. Potential virtual keyboards. And for the software, I plan to use Oculus SDK, Unreal Engine 5, and Blender. Okay, the goal and the case of success. The goal of the study is to create a media to learn more about flow and hyperfocus, create an open source innovative learning media, and deploy the application for researchers and learners worldwide. The case of success is to have a functional VR prototype, being able to integrate the detection of hyperfocus and flow state, and make a good documentation about the study and about the VR prototype. Okay, for the timeline, sorry, I plan to first the 2 first weeks would be to establish the foundation and create the low fidelity prototype, and then to go to development phases where I will code the environment and the task integration, and then to find someone to test it and then refine. Then the integration of the detection would be in the 7th week. And then I will make some finalization. For the week 8 to 10, I plan to test and make to test on multiple people, and then on the 11th weeks, I want to analyze the results, and then the last week will be for the finalization of the study and the documentation of the prototype and the software. So about me, I'm a second year student at Telecom Paris, which is the first computer science engineering school in France. My specialization is in graphic and interactive system. I have a multidisciplinary background with my bachelor in math, computer science, and biology. My interests are in virtual reality. I'm an artistic person because I do some art stuff on Blender, Nomad, and on Procreate. I love programmation, and I have a strong interest in neuroscience, and especially in BCI, even if my project is not directly connected to BCI right now. I have some skill. I have advanced skill in C++ and the form of OpenGL. I have some skills in Blender and in Unreal Engine. Other thing about me is that I'm an ADHD person, and there is a lot of people around me that are neurotypical person. That's why I'm very interested in this kind of subjects. And I was for 2 years a neurotypical private tutor at #1 Scholarity in English, which is a French company that provide some courses for neurotypical people. And for future consideration, I think it would be interesting to integrate some other techniques such as EEG signals or other brain visualization techniques linked with some personalities theories, and maybe create more environment, more tasks to assess other hypotheses, or just to create a platform for learners. Thank you. Here are some of my references, and thank you very much.
Well, thank you, Madama. That was a very good talk. Yeah.
Super cool.
Yeah. Okay. Any questions?
No. Just really interesting. I think it would be I mean, EEG is a great way to go, and I know some of the companies that are doing integrated VR EEG headsets. One of the benefits you have with a lot of the new systems, I'm not sure about the Meta, but a lot of these systems have eye tracking now built in, and it's another interesting signal, like especially for just characterizing differences in behavior, the point of fixation, and how that changes over time can be very informative, and it's also interesting. Oh, sorry, Paula. We're
I don't know.
Anyway, I don't know if that's built into the. I don't know how much that's built into Meta, but it is another really interesting signal, and especially as it relates to flow. I mean, I would love to have pupil dilation along with that, and I sent you a link to the Cog Lab, which is in Paris, and we have a meeting every Wednesday evening that's very much focused on EEG and VR and these kinds of things. So that might be something of interest too. Yeah.
Okay, thank you very much.
Of course.
I take notes.
Yeah. So that was a great talk, and we talk a lot about continuous cognition in the group, and some of the things that we can, basically developing tools for that, because most of the way that people study cognition is generally like trial based, which is good, of course, but like you have this naturalistic aspect, which that's the way we behave, and so understanding that is both difficult to measure because you just have this series of things going on, and then it's also difficult to sort of build a experimental paradigm where you can actually detect differences between one condition and another. So this VR, one reason we like to use VR for that is because it makes it easier to sort of quantify everything, or at least make control the environment enough so that we can get those effects and see like if you, especially some of the things that you're dealing with in flow states, like attention, controlling how attention happens, how it unfolds, being able to add in things maybe like eye tracking, but even just in VR you can track the entire experience, and depending on how you can get what data you can get out of the system, can give you some really interesting data to work with. I think probably doing this in Meta's ecosystem is probably good, because it's easy to access the hardware and to develop, relatively easy to develop things for it. But but there's no, I don't think there's a better platform to do this with, unless you want to strip everything down and go to like something like Monado XR or something. But this definitely is, I think, a good starting point for a lot of this, and I'm really interested to see what we can do with this.
Okay, thank you very much.
Yeah.
Can I ask a question to Adama?
Yes, of course.
Hello, Adama. Thank you for talking. And it's really interesting space. And from the presentation viewpoint, I'm not in the lab disclaimer, so what I say doesn't count towards any of whatever evaluation you're getting out of this. So I'm an observer as well. And as an observer, I think it would be helpful at the beginning to have maybe one slide where you can give an overview of the whole talk, or maybe just one point where you say exactly. You could just, I know it's difficult because you probably haven't worked it out, but give a bit of, try to say, this is what I'm interested in, this is the space I'm looking at, and this is what I'm trying to do, or this is the question that I'm trying to answer. It could help us to focus on whatever comes after, to to have to struggle less is to understand what are you getting at. And in terms of the question that I actually have, is I don't know if it's pertinent and if you have an answer, but I was just curious. Have you, or maybe you have said it and I've missed it, because I also have some kind of attention deficit sometimes. So what kind of parameters will you be evaluating? Did you say that? Well, I mean, when you're going to try your system on people, what are you looking at? Physiological parameters or cognitive parameters, or have you specified that? Have you figured that out? Maybe you have already said it and I missed it.
For the study, because of the cost of the materials to measure the physiological outputs or physiological values, I decided to focus on detecting flow with some behavior with the behavior of the participant while the study, and I also decided to make a study of some questionnaire and quizzes post study to know how because flow is.
When you say behavior, what kind of behavior exactly?
For example, for coding task, if the user has to write some code, I can take some input like such as the speed of writing the code, the
line of code per hour or per minute, something like that. Line of code.
Yes, line of.
Okay, that is that is worth really focusing on. I think also it also gives a limitation to the study in the sense that if you're only taking one parameter and you're gonna get maybe some good results, take some decent measurements on that, but does it actually reflect the overall cognitive response? Not, maybe not, but you got to start somewhere. So that's what I would focus on. I'm not expecting you to give me an answer now, but focus on defining your parameters and maybe diversify them. The line of code is good, but there could be others. And so whatever number of errors that you do per hour, or whatever, how many bugs, how much debugging time, whatever. So you're going to decide that, and I think it's worth defining that a little up front, and then also continuing to iterate and change that as you progress with the project.
Okay, thank you very much.
Yeah, I put a paper in chat that's it's one of the original kind of neurogaming papers that was here in San Francisco. Just as an idea in terms of task, basically how you could add a cognitive task to a continuous performance task. It's down the bottom there.
Oh, right here.
Yeah,
Okay.
yeah. So this is the focus of it was in older adults, but the task was really designed to give people something that they were kind of always doing, and then while they're doing that, you're keeping them in a kind of they've got to keep up with this game as it's changing, right? But then while you're doing that, then you're kind of probing them cognitively, I mean, I thought of your gorilla, spotting the gorilla task, and it's somewhat similar to that in the sense that like it's a multitasking experiment, really.
Okay.
That might be inspirational. Yeah.
Okay, thank you very much for the reference.
Yeah, this is a good paper, and it hits upon this issue that you'll have with attention, which is multitasking, and the sort of splitting of attention versus the focus of attention on one thing versus another thing. That's the sort of the essence of the gorilla task. It's like there's a gorilla walking through the scene and you don't notice it because you're busy looking at people bouncing a basketball around, and it's like you shut out everything else to the exclusion of this one task. So that's something that it'll be interesting. You have to design around sort of to get at that phenomenon, or to sort of make sure that people don't sort of lose focus on some other things going on in the background. It depends on how you want to do it. So there are a lot of ways you can design an environment to with that knowledge in mind to get at some of these things and measure them. And that being said, I think the behavioral measures are much easier to implement than the physiological measures. So I wouldn't worry about the physiological measures right away. I'd worry about basic indicators of behavior and cognition, some measure of like attention, maybe speed or detection or something very simple that you can record and, get a measure that you can compare in between different conditions. So we could have like one condition where, you have one type of stimulus, another where you have another type of stimulus, and then compare those. And then when that works well, then you can maybe go to a physiological measure and see how that works. But to get the test bed down, I think is important. And so that would probably be what we would do this summer. It's one of these projects where it's very open-ended in terms of where you're going to go. So, there are also a lot of kind of the summary of the psychological literature on this, which is to say that, there is some literature, of course, like the paper that we shared with the video game stuff. There's been some work on like video games and attention and how that works, but that's not like there's not a huge literature on it. It's just kind of like, people have tested different things. It's kind of hard to find. It's not been studied to death like just basic attention experiments. It's definitely something that is a kind of a frontier area even today. So definitely this is going to be interesting. How this, we can maybe even write a paper on this, where we, maybe use this environment, or, we get some insight from doing this.
Yeah, and just to add to that, I don't know if you include eye tracking as physio, but like I would say that again, like I can tell you how we, NeuroTech-X can show you how to add eye tracking to headsets, to these systems, if not. But it's such a use, it's an easy thing, but it's also just so important if you really care about flow and attention. Yeah. Like, where is the person actually looking?
Yeah.
And the neurosignals don't actually give you so much as the eye tracking.
Yeah.
Which is interesting. But anyway,
hacking the hardware is going to be a task in and of itself. Like.
I mean, that's way beyond this.
Well, Meta's, got a weird kind of setup. Like you may or may not be able to access the eye tracking data from the headset. Or, Quest 2 doesn't have eye tracking, but if you had like eye tracking, you'd have to kind of, if you had it like it's separate, you'd have to have a separate channel for that to capture it and synchronize it with the stimulus. So
I again, I can go into details, but it's way beyond a 10-week project.
Right. Yeah. But
but if you are interested, NeuroTech-X is the place.
Right.
We can tell you how to synchronize this. But really, really interesting, and
it's
we can brainstorm more about it.
Yeah. So why don't we move on? Thank you again, Adama, for your presentation.
Thank you too.
Yeah. So I had a couple things here. Let's see. We have. I wanted to bring our attention to a couple things. The first is this new post on our Medium. So this is the web Medium, medium.com Orel group. And I put together this post on our embodied intelligence workshop activities. So don't worry about this. So this is just, this is the embodied intelligence workshop that happened in March. So this was an online event. We talked about this in previous meetings, but this is, our contribution to it. So if you go to their website, you have the program from this year, and they have a YouTube channel where they have recorded the talks, the main talks and the main sessions. So you can catch up on those, and there's some really interesting talks there. But it was a very interdisciplinary group. Computational modeling, a lot of soft robotics, some cognitive science, some philosophy and neuroscience, and even some paleontology, and talking about the evolution of embodiment, which was really interesting. It was a really interesting discussion, thinking about like different organisms and how they evolved to live in new environments, and then what the effects of that are, in terms of the anatomy, in terms of their sensory systems and that. And, that obviously has implications for like robotic design and other things. So there are, a lot of people talking about their fields and then kind of crossing into a new sort of frontier area. So that's that, and you can catch up on that. But the stuff we did so I gave a presentation in one of the breakout groups so there were different tracks to the breakout groups. For the self-organized systems track, I presented on some of the work we've done on developmental Bradenburg vehicles. So this is the developmental neurosimulation as a route to embodied morphogenetic inspired intelligence. And I have a recording of the talk here, so you can catch that recording. They didn't record the breakout sessions, but I have a recording of the talk. And it's summarized, by this idea of taking maybe like a very simple developmental form, like a fertilized egg, but it's a computational version of this, which is just a sphere. And then, developing different morphologies, intermediate morphologies to an adult Bradenburg vehicle. So the developmental aspect is where you have this developmental process that shapes the phenotype, but also shapes the little connectome inside, the connections between the sensors and effectors, which are the sensory organs, and the wheels or effectors, the thing that moves it through the environment. And so you can, model those two aspects of the agent and end up with different types of designs for the different kinds of tasks, or just examine how, if you have like an evolutionary program or, some other generative model, you can see what kinds of forms you can create through, different conditions. And so I talk about a lot of that in that talk, and I've included the abstract for each of these ideas. So this was actually a talk that was given in the breakout session. This is the abstract. Now, Jesse and Hussein, they also contributed an abstract, but they didn't give their talks for some scheduling difficulties. But I think, we still have the title and the abstract, so you can look at these, and, hopefully we'll get some, maybe we'll get a paper from Jesse on this, in the future. But I like this idea here. This is something that we've been talking about in Cognition Futures in those meetings. So Jesse's contribution was a mind as a blank, and not like that we have a blank mind, but it's the idea that, we have these metaphors or analogies for the mind being like a computer, or being like a water wheel, or there are a number of things in history that people have used to describe the mind and the brain. So, this is the idea that you can fill in the blank as to your preferred metaphor, but there are a lot of, if you go back into the past or look into the present, people are thinking about it in different ways and using those analogies and those metaphors. So this kind of talks about, he kind of goes through historical analysis. he talks about the dominant analogies and maybe less dominant analogies, and that there's this cross-disciplinary aspect. So if you're doing like robotics, you might think of the mind as differently as if you were doing like anthropology or maybe neuroscience or psychiatry. And so even if you use basically the same metaphor but you have different ways of thinking about what the mind is doing, that's also kind of what he covers in here. So this is really interesting stuff, and I really encourage him to kind of develop this paper, because he has a draft of it, but we need to work on it more, and I'd like to devote some time to that, because I think it would be a really important thing to get out there.
Yeah, and it keeps coming up too. Like I basically wrote like a super extended abstract for another conference too.
Yeah.
I think that's what Bradley recently saw, and I go into detail with more of it, really frame out a lot of things. Some parts were really ambitious and some were not, but those can be broken down into sort of different like components. But I think there is something here, and I think one of the key things that came up during this that I want to. Well, there's a few things. One of them is that the analogy. But part of like one of the undertones or things that were happening while that paper concept idea was being formed or conceived or whatnot, I believe we're going through the Norbert Wiener book, Human Use of Human Beings, and in our discussions we had a discussion group about that, and for a little bit of context, without going too far into things, excuse me. One of the things that was really stayed with me after reading the Norbert book was that Norbert, because of what he was trained in and what he was exposed to, had a very interesting vantage point about how interacting with the current and then forthcoming technologies both would affect like people's, to an extent, embodiments or experience, and then also he was very spoke a lot about how it would affect affect news media, and then affect all these different sort of broader scale cybernetic-ish, regulatory-ish things that were going on. And I think one of the things that kind of comes away from that book that sort of influenced a little bit of this mind thing, which isn't necessarily about cybernetics or Norbert Wiener at all, but was sort of this there's sort of an inescapable zeitgeist of what the analogies can do. Like a little bit more concrete example of this was a long time ago. I started a very, very small blog, and I don't think it really exists anymore. I don't think I put much in it, but it was sort of looking at how technology or the future was conceived across different time periods. Like a somewhat maybe accessible example, especially to the not younger folks watching this. But like if you look at what's the one with Arnold Schwarzenegger, Get Your Ass to Mars?
Total Recall.
Yeah, and just like seeing what that was like in the 80s versus the 80s versus when it was written originally, much earlier. I forget if that was like Philip K. Dick one or not.
Yeah, it is.
But when it was written, when it was first made in the movies, and then there was a more recent one too, and just seeing, this is just a simple example of what was tenable as futuristic, but also able to be understood in that zeitgeist. Like I always remember the scene from the Arnold Schwarzenegger movie where he's pulling the tracker out of his nose.
Yeah.
And it's this giant blinking like 3 centimeter, 2 inch like bulb. Like, oh, I just pulled it out of my nose. And it's like, yeah, because nobody would understand like nanotechnology or like some kind of tracker that you couldn't physically pull out. It had to be that physically. It have to be in that context of in the 80s. That's what you understood. Yeah. And so I think there's a similar perspective to be had on the mind. Like there's a certain, what is a culturally knowable reference or artifact, that you can interpret as that. And I think one of the really cool directions I wanted to go with the paper, that sort of part of work, is that like I would like to break down into sort of I don't know what's classes, but like how interactive is the thing you're making the analogy to, or the comparison to, or the metaphor to? Like, is it just like really early analogies, I think in each were like the mind or the brain, the mind or the essence of someone, or the soul, or whatever was this. It was like something contained in a vase, and then you later in life, or later in history, in different cultures, you have like a piano or an instrument, the water wheel. Now you have a computer. These things that are sort of have different kinds of inputs and outputs, or one-to-many or one-to-one aspects. Like if it's just a bass, it's kind of this thing that just holds it, right? A piano maybe implies a different degree of, things happening, or one person playing the keys and whatever else. And then a computer, and as we discussed last week, computers are great, but there's still much of what we know is based off a very certain type of architecture. Like it's not necessarily the only way to do this or that. And now, now maybe it's, I don't think people are saying the mind is the LLM, but we're to the point where our LLM's conscious and stuff like that too. And it's because that's sort of, part of the magic of ChatGPT isn't necessarily the technology, it's the interface that you can just talk to it. Like you can have the prompt with it, and you can do that thing with it. So that affects, some of what's going on. And like, I think it's pretty safe to say there's never really been a point in time where people are interacting directly with something that has a direct tie to being trained off of so many inputs from like the internet history. Like if anyone asks what's ChatGPT, they say, oh, it's this AI. If you like, a lot of people are just going to stop there. Oh, cool, it's an AI, or oh, that's scary, or whatever. But the next step is like, well, how was it made? Well, it's trained off a bunch of data from a bunch of people that was scraped off of, or stolen, or however you want to go with it. That was taken from many human beings, computationally inputted data things. And so that kind of relationship is sort of a new, I don't want to say it's prevalent, everybody gets it, but like that's different than, even like the printing press or books. There's different sort of macro relationships between the analogy itself. So I think that's sort of where I'd like to go in the future in that. Some of that's a little more, a bit of a reach, but also I think there's really important things in this space about what does it mean? There's some really interesting about how are you comprising an individual, and how discrete are you making that, and how much nuance is it? Is everyone the same kind of piano? Are there different kind of pianos? Are there different, there's sort of this very an side agenda of mine that I'm not necessarily incorporating in this paper or not, but there's a very peculiar, I think the right way to view it is there's a very remarkably peculiar monolithic approach to what a mind is, that has sort of become the dominant thing in many, spaces around it. Like the mind is a blank, and sort of, all things of this class or species do it exactly the same way. And I think particularly for humans, we're entering an area of important diversity of understanding. And like, that's kind of what I've said in the comments for the previous project was like, even like what is like a flow state, or what is a peaceful, like a meditation state? What does being calm look like for a neurodivergent versus a neurotypical person, or someone who has, chronic fatigue versus a certain kind of hyperactivism? And like, are you saying, what is sort of this ideal range for that person, or the normal range for that person? They may look different, even though they have similar, capacities or states or whatever. So that's a whole lot of things. But yeah, I appreciate Bradley for kind of bringing that up again. And and the encouragement on that. I think there's stuff to do there, and I have to
Yeah.
put it towards the top of the list. So thank you for that.
Yeah. And so, yeah, thank you for that. And so then Hussein, of course, did a abstract on, and this is one of his interests in virtual reality. He's interested in spatial cognition and sort of the interactions between like sort of spatial cognition, virtual environments, maybe something like presence or the philosophical implications of that relationship. And so, his focus is on looking at the Morris water maze and looking at sort of the neural substrate of what's going on the Morris water maze is this experiment where you put a rat in a pool and it has to find a platform and go to the platform. And the idea is that you put the rat in at different points and it tries to find the platform, and it's doing a lot of spatial cognition, a lot of spatial rotation to get to that platform. And so sometimes if the rat has a neurological deficit in the hippocampus, you can have deficits in performance. But you can do this also in virtual reality where you have a human agent, a first person view of the pool. You put the agent in the pool and it can find the maze in similar ways. And of course, if you have someone with a cognitive deficit in their hippocampus, they also will not explore the space in the same way as sort of a normal person. And so he's looking at a lot of this sort of hippocampal activity, the behavior in a VR environment, and then the behavior in the Morris water maze task. And so there's a lot going on. And he wrote a paper. He's written a paper on this too. It was for a special issue of a philosophy journal, and it got rejected because it's a very interdisciplinary thing. So he's got a paper on this that we'd like to maybe work on more, and this is his abstract for this talk. So definitely if you have feedback to give him on this, that would be great. But yeah, so this is one of his long-standing interests. So this is what we were doing with embodied intelligence. And then speaking of Hussein today is at this event, and this is the Neurodata Without Borders hackathon, and it's going on this weekend. So this is Neurodata Without Borders is a modeling platform for neuroscience data and simulation, and so this is something that some of the people in OpenWorm have been involved with. I think Neurodata Without Borders is in one of the INCF projects for Google Summer of Code as well. But they have this a hackathon where they're trying to recruit developers and get them to do interesting things. So they have these user training workshops where people use this platform, and they go there and they ask specific questions and they give tutorials, and it's very focused. In a hackathon, you're doing more broad things. You're trying to develop tools and figure out ways to do new things. So the hackathon will enable participants to work intensively on an NWB-related project with the assistance of NWB development team and others in the community. So they want to solve technical problems and foster collaboration amongst developers. So this is hosted by DataJoint, and it looks like they have, I guess this is in person, but so this is just, an event that's going on. So this is good stuff. And it's just like a typical hackathon where you sort of have an introduction to what you're doing. You get project overviews at the beginning of the hackathon, and you work on a project throughout the hackathon. You have breakout discussions, and then you come back together and you give reports on your project. And then you build a roadmap and say, okay, we tackled this issue. We did some things. How does that fit into advancing the roadmap of the project? Let's map that out. And so this is a good opportunity, I think, to at least for Hussein to try to get some experience in this space. And I know that Morgan is a big fan of hackathons, so I don't know if you had anything to say about that.
Well, just of Neurodata Without Borders and its relation to Dandy Archive as well. and it's in an it's part of an ecosystem that maybe I can just post this in Slack, but for tools around, or software projects that are around animal electrophysiology, right?
Yeah.
And so Lauren Frank, again, I've got an obvious San Francisco bias here, but Lauren Frank at UCSF has a project called Spyglass, and it's kind of putting them all together. So tools from Flatiron, which is Simon's foundation, the kind of spike modeling, data joint, neurodata without borders to handle multiple file formats, because a lot of the animal electrophys systems, have proprietary commercial file formats, and a lot of things coming together to create really nice pipelines for this kind of modeling, and again, like really important. There's becoming available a lot of data like those from the Allen Institute, like neuropixel data sets from the Allen Institute, that really do give you the chance to do secondary analyses of things and kind of learn about all the steps, even if that's just the goal, right? Just understanding kind of a modern electrophys pipeline, is great.
Yeah, that's great. And so, that's a project that's, even if you don't attend the hackathon, you can participate in the project, or you can integrate it into things that we're doing, because we're interested in secondary data.
Yeah. So I'll post this to ASU, or if I was free to travel anywhere in the world at any time, I'd be in Phoenix today, or where is ASU? Tempe?
Phoenix. Phoenix.
Yeah, something. I think it's just outside Phoenix. ASU the math department is doing a full day workshop on basically secondary analysis of Allen Institute data. So I think the first half is kind of segmentation based. That's kind of focusing on kind of EM microscopy segmentation, and the second is the kind of neural dynamics and the processing of neuropixel data. And just really cool opportunities there.
Yeah, definitely. So and Paola posted some secondary data from last week, and that's still in the Slack. So we'll have to, maybe next week we'll talk about that more. I don't know. It's it's always kind of an interesting thing to kind of go through and try to work with some secondary data. You can answer a lot of questions with it, and so we'll look into that. And so Paola said, I go to virtual events mostly avoid spatial displacement, but try to attend events when I cross paths. And then Morgan posted this exploring neural data event, and this is at ASU, is Arizona State, and they have this Allen Institute for Neural Dynamics. So this is where they're looking at different types of source data, and you have like microscopy data in this case, and then you segment those data, and then you have these neuropixel probes. You build some sort of lookup table. You have like a way to take the data out of those images, build a table, some sort of computational data structure. Then you can link things like optogenetics and other types of data, and that's how you build like a reference database. And so there's some coding that's required, working with different Python toolboxes, but they're kind of trying to figure out, and this has been ongoing for a while, the best ways to build these pipelines where you have data, raw data, you build a table or some sort of data structure, and then people can query that structure and find things in it. So if I have a question that I want to ask, it could be a psychological question or a philosophical question or a neuroscientific question, I can find data that's, kind of gives me some support for that idea, or I can combine it with other types of data. And having that accessible pipeline makes that easier because it's very hard to sort of, put data sets together or get insights from different sources of data. So the easier you make that process of, standardizing everything, making it easy to maybe graph out, it makes those then later comparisons easier. So, this is all part of a sort of a larger agenda. So yeah, that we can revisit that later. Now Paola posted an article during Adama's talk, and this was the paper on from Brain Science. This is actually on our earlier discussion. Yeah. The effects of trauma with or without PTSD.
Sorry, because I'm multitasking all the time. So I was listening. 1 year I was listening to Adama's talk, and with another part of my brain, I was following on the previous talk.
Okay.
And this is how I end up, being confused. But that was an important point. Thank you for picking up on that, Brad. Is it clear what this link is?
Yeah. So this is actually from the earlier discussion we had on organoids and looking at disease, and then saying, we spend a lot of effort on learning sort of the molecular bases of diseases, and the NIH, of course, funds a lot of that, and then they fund some work on, like looking at sort of trauma and things like that. But what's the balance there that would be optimal for learning about diseases and their multitude causes? This is an interesting paper. This talks about trauma, getting a handle on what trauma is, and then looking at transgenerational DNA methylation alterations. So this is where if you experience trauma in your lifetime, your offspring or your children will maybe inherit certain, some epigenetic imprint of that trauma. And so there have been a number of studies where they've shown that this can be passed down over maybe 2 or 3 generations, that, it can, like if you have situations where, you have famine, and they've done this with, like after World War II, they did this in Europe, looking at different populations of people where they're, people have suffered famine, and then their children inherited these markers, and so there's this literature on this. People have also looked at transgenerational methylation alterations in C. elegans as a model organism, and in C. elegans you can actually do a lot of really interesting manipulations with looking at that lead to these transgenerational inheritance patterns. So they've been able to actually follow that out in C. elegans up to about 10 generations, where, and the generations, of course, are shorter.
It follows on Morgan's, I think, paper as well. Is that right, Morgan, what you just shared with us?
Yeah, absolutely. So, this is kind of what I was, I think the paper that I posted in computational psychiatry is, this would be kind of one of the physiological mechanisms that they're interested in terms of the like it's really a reduced, a review of all the different physiological processes that underlie trauma, and of course.
I just pasted another paper following on the same point that we were discussing before. That so there is a widely held view that there is no correlation between emotional stress and cancer. But this article at Harvard says that there is link. That is just pointing to following the discussion. We're studying cancer at genetic level, which is fine. It has its own merits that work. But if you really want to understand cancer, maybe you have to look at, how people live in the world, the way they, what they eat, the kind of information they consume, the kind of psychological conditioning they're under. And this paper here, the Harvard one, points in that direction, and I think there's not enough data on that.
Yeah.
Yeah, no,
Just to
for,
that point.
for sure.
Paper.
I think so again, like this is very, very close to my own interests in terms of psychiatry. I suggest that comprehensive oncology practices are kind of the best model for psychiatry, and I think you'll see pretty much all cancer programs certainly comprehensive ones include like what can we do to boost the patient's well-being, like does the patient have caregivers, does the patient how what kind of emotional support, not just for their sticking with treatment, so to speak, but really to boost the treatment itself, like without necessarily understanding the physiological mechanisms behind it. They somebody who's going to go through cancer treatment, they don't want them to be doing that alone, because they get better outcomes when the people are supported and the person is practicing time for themselves and self-care. Really important.
That's super duper relevant. I don't want to interrupt by talking about DSM stuff project, but like Jen, who is working on that project, is doing a really deep dive into a documentary about I forget what it was. It's something about anxiety was in the title, but it was looking at specific practices into that space, and what discussions led us down to. And it's funny because we were watching an older television show that was sort of like a really early sort of presentation of PTSD at the time, and between that TV show and the actual documentary, which is like by real serious researchers or people who have like written papers and published on why they're doing this, it really comes back to the point that Morgan just said about that, and sort of how there's treatment and there's actual biological, neurobiological stuff happening in play at that cellular level, or that organ and internal like biological level. And there's sort of the practice, and there's also like one of the things we were talking about was trying to come up with some terminology around sort of like performative recovery or masking during recovery. To say, oh, well, I'm okay, I'm fine, and then or the authority figure, the person, the doctor or the spouse, whoever you care about, is present. So the pressure to put a certain way too, and how that's related to sort of like scaffolding needs, or like these like not just sort of the key points, but the transition, and how that reflects the a lot of feelings about agency, a lot of feelings about what can and can't be done in situations and so on. So that's a really interesting point that's coming up as we're looking at that too.
Yeah. I whenever I want to make this point, really clear, I think the studies on long-term care workers and the heal time, giving them a small directed cut and how long it takes to heal, versus people in different jobs, like this is getting at that, stress, lack of sleep, other things have, like without necessarily understanding all the process, it's like everybody understands, hey, if it takes 3 times longer to heal a cut that you've, a surgical cut you've given them, that there's something really going on that's important to understand.
Yeah, definitely.
Yeah. So yeah, this paper, this is about sort of stress and trauma and how those things kind of relate to both psychological state and some of the things that are going on in the metabolome and the expression of genes. This is a nice paper, but I did want to review a little bit of the Brain Organoids channel and maybe do a couple of selected things in that, just because we don't have very much time left. So let me do that. Let's see. Let me do that, and we have a lot of things in the Slack. So please check out the channels at your convenience. So I mean, in this channel, we do have things on organoids, we have things on stem cells, we have things on like biological modeling, and then other things that maybe relate to psychiatry. We have the computational psychiatry channel here, which has a lot. We went through this before, but this is a separate channel.
But it does reflect my bias.
Well, yeah, you have a certain approach to it. So I mean, so we have like we have videos. So this is mobilizing mathematics for the fight against cancer, and this is biological modeling or mathematical modeling on cancer. We talked about like the difficulties in defining cancer states, or at least different types of cancer, and modeling this, of course, is some physiological stuff. There's psychological aspects and cultural aspects, and all these things that come together in. And so this is a video though for this. Yeah.
But just getting at certainly when I'm posting these kinds of cancer research cancer research material in this channel, it's getting at the kind of leaven. Cancer is a breakdown of collective collective goals of multicellularity, right?
Okay.
And like I'm definitely going for kind of a computational neuroscience approach to organoids, but there's a lot, again, like oncology is the place I see as the real interdisciplinary ground of medicine right now, especially for this kind of again, like tissue organization.
Yeah, that's a good point. There is a lot you can learn from if you want to study the sort of the behavior of, or rather the dysfunction of multicellular systems. Cancer is a good model for that because you can get a lot of information. You can do studies in culture. You can do studies kind of tumors and things like that are
as well as again the kind of the interesting interplay between the genetic expression that you see in any particular cancer because of the tissue that it's coming from, and how that limits it, how that changes it, how that makes it more deadly, all of these things, understanding that relative to the what they would call the tumor microenvironment and things like that. Yeah, it's really it's where the reductionist approach is kind of best expressed right now.
Yeah. This paper, a feedback-driven Internet of Things microfluidic.
If I'm right, I think that's a Katerina paper too.
Okay, this one, yeah. So this is Katerina, who's we've talked about before. She's at UC Santa Cruz in their group, organoid group, or maybe she.
It's our first author paper. Yeah.
So this is a feedback-driven Internet of Things microfluidic electrophysiology and imaging platform. So this is like for brain organoid studies, just like kind of how you would. We talked about this actually, where they want to do Internet of Things because they want to be able to use it for education. They want to have like organoids in different places, and you can learn about organoids and the different measures and doing it like that.
Yeah, UCSC is running a mathematics of the mind course in bioengineering right now that is that has using Jupyter notebooks to control the experiments and process data. Yeah, it's really cool. I'll drop a link for that.
Okay, yeah. Then there's researchers design foundation AI models for use in pathology. So this is these foundational models that we've talked about, but using it for looking at pathological signatures and microscopy images. So, from something like this, can we find a signature of pathology? This would be very useful for doctors, and a lot of doctors don't have like high-end AI knowledge. So having those foundation models is very valuable for that discipline.
Yep.
This paper here is we talked about this paper. I think this was this in this group or in DevoWorm?
I think you had it in DevoWorm, and I loved it.
Okay, yeah. So this is just quickly, if you want to learn more about this paper, it was in I think last week's DevoWorm meeting. Well, we talked about this paper where they were looking at transcriptional networks and using techniques to learn what those are. And so it's really interesting work. And Adelson Motter is a network science person. So this is all, this is kind of like the merging of computational network science and biology. Okay, and then this stuff about organoid intelligence, where like they're, we've expressed criticism of this or skepticism, where you have these organoids that people are talking about how they express intelligent behavior, because you can grow an organoid in a dish and get electrophysiological signals and train it to play something like Pong, which is like basically getting those outputs and stimulating the organoid so that it produces outputs that are sort of able to do a very simple control task.
I will say though, Thomas Harding, is very grounded toxicologist.
Oh yeah.
I do feel like that this. I don't know how much the marketing for this title came from. I think what is this, Charles River?
Yeah.
Associate. Versus, he's doing great stuff, and Johns Hopkins is in particular is really pushing this. I don't want to say campus wide, but across. I think he's in the public health department.
Yeah.
And it's really like providing this viable alternative to working with animals, but again, like bringing everything that in vitro experiments allow you, right? This just like kind of full control, unprecedented control over kind of the physiology,
right?
That really is remarkable. I forget how much he actually goes into kind of neuromorphic computing, but I do want to say one thing, because just about that I think is interesting, which is Intel's got some big neuromorphic computing announcements. I liked what some people were saying, which was just like, I bet you we will see neuromorphic computing become a thing, in the sense that like actually taking some market and some of the compute resources that are the focus of our computing kind of development, because it actually provides real benefits as opposed to quantum computing.
Yeah.
which a lot of people are, like, again,
very hypey. Yes.
I love the comments on Twitter, but basically people are calling somewhat cult-like, and it's like it's always coming, and I felt like the DARPA DSO days was kind of like that. Like there was a lot of talk about quantum computers are going to change this, they're going to change that, and they had a great speaker who focused on quantum logic gates and stuff, but at the same time, it's just like this is so not ready for any kind of applications.
Right.
Whereas Cerebrus as a chip is able to do some incredible things with silicon now with large language models, which have a variety of use cases. Just want to put that out there as like, I really do think that the neuromorphic computing case has a strength to it that doesn't that it is more matched by reality than quantum computing.
Yeah, I mean, yeah, that's the thing about the thing about quantum computing is the benchmarks are very different from like a classical computer. So even if they, you can develop them to do things, you have to figure out how to write the software. You have to figure out what the benchmarks mean. You have to then build a sort of an algorithm that can be used. So for LLMs, you have to figure out how to do that and how to benchmark it in the benchmarks of the quantum computer and all that.
Right. And any real quantum computer is obviously going to be a hybrid.
Yeah.
Right.
And so with neuromorphic computing, I'm not familiar with the benchmarks for that. Is that something that's...
Yeah, well, so there's varied, right? Because, you have so many, it's applicable in so many different ways, right? So, like quantum too, but the people focused on memory, people focused on power efficiency, people focused on, actual compute in terms of how many, I guess they don't use flops, but there's a new, silicon neuromorphic supercomputer that just came online in Australia. I'm blanking on its name right now. Some of the kinds of things that we'd seen previously with like brain scales and Spinnaker as part of the human brain project neuromorphic computing platform. But the benchmarks are there, and it's another nice project that's coming out of UCSC in terms of open neuromorphic. So, and like the open neuromorphic speaker this week was actually somebody that works in Milford's lab and or works together with kind of a well-known robotics lab in Australia.
Yeah.
And like a lot of people are looking at this as, again, for applications where you don't have access to the cloud, so like how it kind of relates to tiny ML kinds of things in terms of, can we make adaptive systems that don't necessarily remote settings, drones, rovers, those kinds of things.
Yeah. So yeah, it's very interesting stuff. Let's see. This is from Marion Liebert Publications from Michael Levin in Bioelectricity Journal. So this is Bioelectricity, an interdisciplinary bridge into the future. Talks about, so he runs his journal Bioelectricity. That's sort of one of his main things. Yeah. And they just started this journal like 5 years ago. So they're doing a sort of an overview of what they've learned. They've really kind of done a lot of sort of AI related things, and kind of getting at this bioelectric code. So talking about how machine learning tools are need to be not only predictive but have an understanding, and you need to be able to map between bioelectric states and downstream transcriptional anatomical outcomes. So he talks, he gives a lot of information about this. He also talks about diverse intelligences, which is a multidisciplinary effort to understand how evolution exploits the properties of biophysics to implement memory, integrated information processing, prediction, and decision making on a wide range of substrates. So it doesn't have to be at the substrate of like our brains. It could be like in an organoid culture, or it could be in somatic cells. It could be in a lot of substrates, even silicon or something else more exotic, like slime molds, as we've talked about in previous meetings. Bioelectricity is well known to neuroscientists as the cognitive glue, which enables a collection of neural cells to develop not only efficient behavior, but also an inner perspective that feels like a unified emergent self. And so and then these are not all, talking about different techniques like optogenetics and CRISPR to use and also the new discipline of cancer neuroscience as sort of like the measures of bioelectricity and the model systems of bioelectricity. So he talks about, artificial intelligence. He talks about, some of these diverse intelligence perspectives. He talks about the sort of the experimental test beds for this. So this is just kind of mapping out a field and saying, where we've made contributions. It's really interesting stuff. And that's really kind of, this bioelectricity stuff. He talks about it. His basic hypothesis is that neurons have action potentials and they have ion channels that produce some sort of electrical current. And then not only neurons do that, but other cells as well. So you have, things, different other cell types in the body that have electrical potentials that have ion channels, or they don't produce action potentials, but they produce these potentials that are not action potential like, but they're electrical fields. They produce electrical fields. And so all these cells that have these potentials, they produce electrical fields and they can communicate with one another. And this, contributes not only to cognition, but it contributes to morphogenesis and other things. So this is all kind of his sort of approach. And this goes back in biology. There's a history of people working with bioelectricity. Of course, it goes back to Volta and some of the work he did with muscle. So, muscle is an electroactive set of cells that, produce, power outputs and force. And so, even Darwin experimented with some of the aspects of bioelectricity and the effects on behavior and things like that. So Levin is following up in that tradition, and he's really kind of trying to build a theory of intelligence around it. So that's where they're coming from with that journal.
It's always cool to say that Darwin did look at these things, and if I write, one of his interests was in plants' use of electricity.
Oh, yeah.
Yeah.
So it goes beyond animals. You see these bioelectric potentials in plants and even in bacteria, which is it's nice for a kind of a unified model of intelligence. Although I don't know, that's a little controversial, and there are people I've seen pushback on in that aspect of it. But anyways, so yeah. A lot of stuff here. This is label 3 3D live cell imaging in mouse embryo, from molecules to multi-cell modules.
Sometimes I put things in the channel that would be useful in terms of the technology or hardware that would be really vital for the analysis pipelines as well as the culture tools.
Yeah. So this is for molecules to multi-cell modules. This is another analytical thing. This is the multimodality cell segmentation challenge towards universal solutions. This is, in our DevoWorm group, we do a lot of work with cell segmentation, and we've actually done some things. We have some data sets that we work with that are based on the cell segmentation challenge. This is actually the multimodality cell segmentation challenge.
Yeah.
So this is where you're looking at microscopy images, and you have information in those images. Sometimes they stain the cells with markers, and the idea is to pull out as much information as you can. So sometimes you want to segment the cells, the cell boundaries. Sometimes you want to identify what's being expressed in the nucleus. You can do a lot of those things and get those data out of the image and turn it into numbers so you can analyze it in different ways. And so what they're doing here is they're saying that, there are different imaging modalities, like different ways to capture imaging information, different information you can capture in those images, and then bring those together into a model of what's happening in the thing you're trying to image. So, go ahead.
No, I was just going to ask. I've seen a lot of papers from Bo Wang, and I was just wondering where that person's located.
He's in Toronto.
Toronto.
Yeah.
Okay, Toronto. Yeah.
So he's done a lot of stuff. Yeah, he's like.
I think he's on some of the. Is it the Sec Sec GPT? Maybe.
Yeah. Maybe.
It's something like that. Like I don't know if that's actually it's
the same guy. Yeah.
But I think that might be the same guy.
All right. Yeah. So this is like interesting with respect to DevoWorm. They're doing a lot of things with segmentation and using different algorithms, and then these challenges are basically where you give teams a bunch of data and they try different techniques to try to get the best results. So this is. Let's see if they mention this here. They mentioned the cell tracking challenge, the CTC, the data science bull challenge, the colon nuclei identification and counting challenge, conic. That's very specific, but it's a very specific task we want people to solve. So, and then of course we have cell pose, which is sort of a model that we can, an example of a model that we can use to do a lot of these kind of analyses. So Cell Pose 2.0 demonstrates the efficacy of adapting a pre-trained model to new images. So you can take a relatively small number of images, 500 to 1,000 annotated images, and then just be able to simulate or get analysis that is very good and has a high level of performance. Usually you have to use many more images to train a model. So this is, the challenge here is to make compact models that are very highly predictive, that give us some of this flexibility, especially with multimodal data sets and bringing them together and having a high level of performance. So this is definitely something, if you're interested in that area, to read.
And super important, in the microscopy world, these are kind of the analysis problems that people need solutions for, software solutions for.
Yeah.
Or is like really vital to turning images into insight. Yeah.
Oh yeah. So let's see. This is interesting paper on Optogenetics-integrated gut organ culture systems connecting enteric neurons, dynamics, and gut homeostasis. So this is again on these, sort of peripheral nervous systems models. So this is of the enteric nervous system. This is where you're building a gut organ culture system. You're building optogenetics into that, so you can. And so what optogenetics allows you to do is, you can take you knock in a genetic element that has sort of a fluorescent protein in it, and then you can shine a certain frequency of light on the cells, and it activates that fluorescent protein. So the protein is expressed and it's fluorescent in the image when you take it, and so you can do this in a controlled way. So you can turn on a certain frequency of light, it stimulates the cells, it gives you that signal when you want it. And so what they're doing here is they're building a model in culture of the enteric nervous system. And the enteric nervous system is important for understanding the gut and the microbiota and the immune system and some of the neural aspects of it, neural control. So this is a really interesting system to work with because you have a lot of different subsystems coming together. And then this allows us, so we present a novel optogenetics-integrated gut organ culture system that enables real-time whole tissue stimulation of specific ENS lineages, which is the enteric nervous system, allowing for detailed analysis of their functional impact. We demonstrate that optogenetic activation of enteric cholinergic neurons rapidly modulates intestinal physiology. And so you can look at neuronal firing patterns and how they modulate neuroimmunological gene expression and the epithelial barrier integrity. Basically, the way the organ functions and some of the gene expression patterns that lead to interactions with the immune system and with other things. Like nervous system, immune system, and those intersections. And so diverse enteric neuronal lineages, which are the cells in the enteric nervous system, they form different cell types. They exert distinct regulatory roles. So cholinergic activation promotes gene sets associated with type 2 immunity. Tachycholinergic enteric neurons differentially control mucosal defense programs, and luminal induction of the intermodulatory bacterium C. ramosum significantly remodeled cholinergic-induced neuroimmunological transcription. So there are all these interactions between gut bacteria, the immune system, and the function of neurons in that system. So this is really interesting stuff, and you can learn a lot from these kind of isolated systems, but that have some biological integration. Okay, so I'm going to finish up for this. I don't want to get too much into the rest of this. We have some really interesting articles on methods, on techniques, brain stimulation, some other things here. So, yeah, I won't get into this, but go through join the channel if you're interested. Go through the papers if you're interested. Let me finish up by looking at the chat, because we had a lot of stuff in the chat here. So yeah, we had, we talked about this paper here. Jess's major dovetail with the DSM project. The trauma paper, I don't know if it's in here.
Yeah, see, Paula got it for me,
Okay.
so that was resolved. That's all good. Thank you
Good.
for that.
And then this is the paper. Okay. Let's see. Major dovetail. Oh, she's asking about the dovetail. So yeah, you talked about the DSM project and how, they're looking at reinterpreting the DSM. So, the DSM is, of course, has a lot of problems, and they're looking at ways to reinterpret the DSM, especially in terms of like neurodiversity and things like that. And, that's a project that Jesse's working on with Jen Jiang, I think. And and so they haven't really, we haven't presented to the group. That might be a good thing to do soon, if you're ready. I don't know.
Soon. We need to make things like interpretable for the group, but there's stuff happening into it. Yes.
Okay.
There's a few other things I'd like to really quickly mention before we close for today.
Okay.
But I don't know if you want me to do that now or you want to say something else
Oh
first.
no, I think Adama had to leave, so thank you, Adama, for your presentation and coming today. And
I think Sarah was here briefly.
yeah, Sarah was here, so thank you, Sarah, as well for attending.
Okay, I'll really quickly share my screen because this page is going to go through a few things just to sort of put it on the record as if we're
okay.
I've been watching a lot of old Perry Mason shows, like the old school ones, and so I'll make sure that the court has this on record for the time being. We could reintroduce it later. Anyway, I think the easiest way to do it will be to just go through some of this. I'm going to Oxford and London next week, so let's chat about that. I'll be there for the inclusive gaming event, which I talked about a little bit in the meeting Wednesday. I'll say more about that in Slack, but there's an Oxford Internet Institute. And this should be a lot of fun. We're going there for plot twisters. We're still deciding who else is going to be there. I wasn't going to go at all. I will be there. Talk about plot twisters, inclusive game. It'll be a lot of fun. And again, I'm just going to go really quickly through everything else here. But more ahead on that. This is the bit about teaching philosophy of
Yes.
science, which I'm on board with. Okay. Some interesting initiatives coming out at my home university, San Diego. Like, I'm not going to go to this article, but mostly agent systems, digital twins, and some oscillation stuff is getting like big time funding from basically the Defense Department at San Diego. San was really successful. I just want to plug them. Just so many good things are coming from that. We've had speakers talk about it. The pictures are coming out from it. But a lot of people, it's been hard to kind of get afterglow from it because it's been hard to collect everything, but people are using LinkedIn really well this year, so we can see all these pictures and really fun comments from a bunch of different people. The poster sessions, this word cloud. There's a lot of really fun thing here, so check out New York Celebration of Women in Computing for that. I was a co-chair like officially this year, and I've been with them for a long time. I didn't get to go in person, but I helped out from afar for the event. But we're looking forward to next year too. That was really successful. So congratulations to everybody involved with that. And also shout out to we didn't present anything this year with a lab at RL, but we probably will next year. And thank you for the continued sort of interest and support for when we did present there from the community. So thanks for that. Levins and his multiple layers of scale, always good stuff. I was at this big event, and I'll talk about that more next time, but basically it was a really big insight into Boston AI tech and AI scene. There we go. Here's my cool thing at the Media Lab. One thing I'll say about it, it was so weird because the Media Lab is not built for a big event, and the Media Lab is a really, really quirky space. Like there's all these weird, I don't know if I have any good pictures here, but there's all these like weird like fluid interfaces lab and everything else going on there with like quirky projects and really fun and exciting. Yeah, you kind of see, you know, but these pictures make it look like it was this big conference venue, which it is. There's good rooms for it, but you can kind of almost see like in the back here, like the heart of the Media Lab is these quirky lab spaces that are doing really interesting and novel things. And so to have this big tech, big AI, you know, energy around it, and you have like Stephen Wolfram and Jan LeCun, and Lex Friedman was there, and all these like big names talking about stuff too, and there's Joshua Bach and stuff. Like it was such an interesting mix of, oh, this makes perfect sense for the Media Lab at MIT and making things to products and big tech, and also like kind of very humorously out of place. So it was a fun time there, and you get a lot of like very coded language about responsible AI versus big tech and optimism and how to talk about it. So I'm writing about that, so more of that for next time. And then lastly, I wanted to finish on really great discussion. Oh, this is kind of the brain initiative funding getting cut. Is kind of a big thing for the neuro stuff, which sucks. As we know, I love my whiteboard spaces. RIP to the Future of Humanity Institute. Perhaps controversy with Bostrom brought it down, but Andrew Steinberg always did a lot of great stuff, and he kind of wrote some perspectives on it. Some stuff from Johan. And I'm going to close on saying really, really, really great discussion finishing up the allostatic, the interoception modeling, allostasis control paper from Eli was first author, and then Elisa Feldman Barrett's group at I think Northeastern, right?
Yeah.
But yeah, and then next week we're going to dive into Tom Froese and eruption theory and kind of what he's up to with that. He's just been releasing a bunch of stuff the last week, months at some like big official events. So it's exciting times for that, and looking forward to the cybernetics reading group, which didn't meet this week, but more next week. I think we're talking about Levin or something else there. I forget, but this was a nice discussion for finishing this paper, getting a sense of what they were doing, getting to the meat of the paper, having a little bit better discussion. I'm trying to improve that too. Things are going well, but a lot of people were excited about that and excited about the work ahead. So that's my super rapid recap to close this week, to get it on the record and talk more about it next time.
Well, that sounds great. Thanks for the update. And again, thanks for Paula for attending, and Morgan and Jesse as usual. And yeah, the great conversations today. And let's keep talking in the Slack and going through the Slack, making sure that we kind of digest things that get posted there. And yeah, and then of course we'll have our meetings this week. We'll have our Cognition Futures, and I think we'll also have a cybernetics meeting as well as DevoWorm. We'll be starting up our open source meetings soon.
Yeah, they're coming soon.
Yeah.
They're almost here.
So we'll
Already
see.
Here.
All right. And then, yeah. So thank you. Have a good week.
All right. Take care, everybody.
Take care.
Take care. Bye bye.
Bye.